My name is Chris Doe. I'm the primary investigator or faculty mentor that runs the lab here. And I'm a professor of biology. I'm also an associate director of the Institute of Neuroscience. In the Doe Lab, we are interested in a big overarching question of how do you fix a broken nervous system. And to achieve those goals, we feel like we need to understand how the brain is built to begin with so that we can use those techniques and skills to rebuild it when it's broken, either by injury or disease. And so our lab runs on multiple levels. We work on stem cells, neural stem cells. How do stem cells regenerate themselves? How do stem cells produce different types of neurons? We're very excited about the work we're doing in Drosophila neural stem cells. We also work on how neurons form circuits. How do they find their partners to form a circuit to allow motor behavior or even cognition?
I joined the Doe Lab back in spring in 2017 and had the opportunity to do some science with an older graduate student at the time, and was really introduced to the power of dissecting questions about nervous system development using the fruit fly as a model system. So I just really fell in love with the ability to look deeply into the brain with single cell, single connection resolution, to be able to observe and then ask questions about how the brain comes together during development. The question that I'm really interested in answering is how cells in the brain wire up with tremendous specificity in order to enable an animal to conduct all of the behaviors that it needs to do in order to survive.
My name is Sonia. I'm a senior. I've been working in the Doe Lab since just before I was a freshman, so about 4 years now. Currently I'm working with Dr. Sarah Ackerman on a project to identify the role of glial mitochondria in the development of Drosophila larvae, in the hopes of transferring that data and information to humans in terms of an ALS type phenotype. And we're hoping that we can kind of elucidate some of the underlying mechanisms for why ALS progresses the way it does.
Typically, an undergraduate in the lab would get a dedicated one-on-one mentor and then learn how to do the science in the lab. And this is super important because classes are only part of your education. What we really need to do is apply that in the lab as a scientist.
Joining a research lab as an undergraduate was really important and really formative. As a senior graduate student, I've been able to train undergraduates and junior graduate students. Those experiences have been really powerful.
The Doe Lab seemed to offer a lot of opportunities for me to get into my own research after some training, and it seemed to have a lot of opportunities for me to explore my own passion for science. I would say it's been extremely impactful for me. I mean, it changed my life completely. Through my work in the Doe Lab, I found that my passion is more for benchwork science and working with mechanisms. Everything's changed from when I was a freshman, that's for sure.
The key, I think, for me was getting into the lab at the beginning instead of in the classroom, and that was formative. And I think most people who are successful scientists, whether they're in industry or biotech or academia, they all had a research lab experience that got them excited. So I would encourage anybody who's interested in neural stem cells, neural circuits, behavior, fruit flies in general. They do have brains. I would encourage anybody just to email me. And we're always looking for motivated, talented undergraduates. Thank you.